So it is getting live streamed in YouTube now.
Okay. Dr. Reed probably left. He'll join again.
He has left.
Yeah, he'll join again.
Okay, please, Enakshi, if you can help him.
Yeah, sure. Yeah.
And actually, it will be good if you can ask for the presentation from your boss. You can present it, and it will be better, I think.
I can't understand.
If the presentation is with you, then you can present the screen, and Dr. Reed will
Dr. Reed can
give the talk.
Give the talk.
Okay, but
If
then
it is possible, then he has to mail it. Then he has to mail it to. He has to mail it to you first, and then you will be in the meeting, and you can hear and subsequently navigate the screens.
No, the problem is I don't know what is like exactly what are the sequences of his slides and when to change. So.
Okay.
Then talk to Dr. Reed. Nakshi, please talk to Dr. Reed.
Yes. Hello. Hi, sorry, Dr. Reed. I think you got disconnected. Can you join in back? Yeah. Is there a problem? Okay.
Yeah, Dr. Reed is joining. Dr. Reed is joining.
Okay.
Okay, he has joined.
Yeah, you just joined. Okay, thank you.
So within 1 minute we will start. Okay, Shilajit? Okay, Harsha?
Yeah.
It's 6:59 now, so we'll start at .7.
I think Dr. Reed has muted his microphone.
Yeah.
Unmuted.
Okay, sir.
Yeah.
Now, can you see the presentation?
Presentation? No,
No, we are not seeing it. Previously it was visible.
The same way which it is.
Shilajit, please help Dr. Reed.
It's a button called present now in the right hand side corner. Present now.
Bottom right hand corner, sir.
Bottom right hand corner. Yeah, it's okay. Yeah, it's okay.
It's done.
Yeah, it's okay. Exactly.
Fine.
Yeah, fine. Very fine.
And it's full screen mode also, so it's okay.
Yeah, it's okay.
Okay. And can you see me?
No.
Okay, but that's a problem because I want you to see me. Anyway, do you want to begin?
If I unpin you, then you will be visible. But in that case, your presentation screen will be minimized in a small scale.
No, it's okay. You don't have to see me. So first and foremost, thank you for the opportunity to speak, Dr. Das.
Thank you.
I appreciate it. And who am I speaking with? Who am I speaking with?
There is a schedule protocol already set by us. So we'll start that. Our anchor is Miss Harsha Sharma, who is doing postgraduate in our department. So Harsha will anchor the whole program. There will be a short welcome address by Dr. Shompa Devnath of our department. Then Enakshi will introduce you to the audience. And after your talk, there will be a question answer session that will be conducted by one of our MSc postgraduate student, Miss Shatrika Das, and vote of thanks will be given by Dr. Shilajit Borua. So before you start your talk, may I start the proceeding?
Sure.
Okay, sir. Thank you. So, Harsha, it's already 7 p.m. So you may start.
Sanjeev, you are requested to start recording.
Okay.
Sir, so then I'm not turning on my camera.
1 minute. I'm starting the recording. It will take a little bit time. Yeah, now it's getting recorded. So, Harsha, please start. No, you don't need to open your camera. You just start.
A very good evening to everyone joining us for the second session of International Webinar Lecture Series in Search of Origin, Prevention and Possible Cure for COVID-19, organized by the Department of Microbiology in collaboration with Internal Quality Assurance Cell, Vijaygarh Jyotish College. For this session, we have Dr. St. Patrick Reed, Assistant Professor, Department of Pathology and Microbiology, University of Nebraska Medical Center, Omaha, Nebraska, USA, discussing therapeutic implications of understanding SARS-CoV-2 infection from the inside and out. Before we begin this session, I would like to remind the participants that the YouTube live chat box is strictly for posting questions that will be later discussed in a dedicated question and answer session. Please refrain from casual conversation. And the feedback link will be provided in due time, so kindly do not flood our chat box requesting the same. Now, I would request Dr. Sompa Devnath, Assistant Professor, Department of Microbiology and Joint Coordinator of Internal Quality Assurance Cell, Vijaygarh Jyotish College, to begin today's session with her welcome address.
Thank you, Harsha. Very good morning to our honorable speaker, Dr. Reed. And good evening, all of our participant faculties, students, and my colleagues. The Department of Microbiology, in collaboration with Internal Quality Assurance Cell of Vijaygarh Jyotish College, has organized international webinar on In Search of Origin, Prevention and Possible Cure for COVID-19. This is the second session. As our principal madam is unable to join this program due to technical issue, on behalf of Vijaygarh Jyotish College and Internal Quality Assurance Cell, I, Dr. Shompa Devnath, would like to welcome our honorable speaker, our participant faculties, students, and my colleagues present here. Let us enjoy the talk, join the talk. Thank you.
Thank you, ma'am. Now, to formally introduce our speaker this evening, I adjure Enakshi Roy, a PhD student developing models for studying chikungunya virus pathogenesis in Dr. Reed's laboratory at UNMC, and an ex-student of our college. Over to you, Ms. Roy.
Hello, everyone. My name is Enakshi Roy. I'm currently a PhD student at University of Nebraska Medical Center and an alumna at Vijaygarh Jyotish College. Today, I have the great pleasure of introducing Dr. St. Patrick Reed as the invited speaker for this international webinar lecture series. Dr. Reed was born in Jamaica. He did his BS from University of Rochester in New York and his MS from University of Georgia in Athens. He completed his PhD in the year 2007 from Mount Sinai School of Biological Sciences, New York. He then did his postdoctoral research from Mount Sinai and then from Lyon for France. He was a research fellow at the Division of Integrated Toxicology, United States Army Medical Research Institute of Infectious Diseases, Maryland. Dr. Reed is presently an assistant professor in Department of Pathology and Microbiology at University of Nebraska Medical Center, Omaha, USA. As a virologist at UNMC, he conducts and supervises fundamental research on different viruses, including Ebola, chikungunya, Zika, etc. He is my supervisor in my PhD program. He also supervises other MS and PhD students in his lab. He has authored and co-authored in several peer-reviewed scientific publications. As an expert in virology, he is presently coordinating and conducting several projects on SARS coronavirus 2 at UNMC. He has recently published a review article on molecular aspects of COVID-19 differential pathogenesis. He has also submitted a manuscript on aerosol and surface transmission potential of SARS-CoV-2, where he detected viral contamination among air and surface samples, indicating that SARS-CoV-2 may spread through both direct as well as indirect mechanisms, thereby supporting the use of airborne isolation precautions when caring for COVID-19 patients. He has recently received a COVID rapid response grant from UNMC. Today, his topic for presentation is therapeutic implications of understanding SARS coronavirus 2 infection from the inside and out. With that, please join me in welcoming Dr. Reed with a virtual big round of applause. Dr. Reed.
Thank you very much, Anakshi. That was a fantastic introduction. So thank you guys very much for the invitation to be here. Like Anakshi said, my name is St. Patrick Reed. Can you hear me well? Everyone's good?
Yes.
Okay, good. So what I would like to do is go through a little bit of the work that I'm trying to do and that we've been conducting on SARS-CoV-2. And first and foremost, a lot of the work that we do is in complete collaboration with a lot of other individuals. And so we have to make it very clear that it's not just us. There's a lot of people that are involved. And so with that said, how do I scroll down? Okay, so can you see the next slide? Are we good?
Yes.
So it's important to know that before COVID-19, there was a focus of the lab on chikungunya virus as well as Ebola virus, because we know that we had other things that we prioritize. And so in particular, one of the things that's of interest to me and that Anakshi's been working on is looking at how bone pathology works during chikungunya virus infection. And so one of the things we've been interested in is understanding what underlies the bone pathology. And so to do that, we've been looking at osteogenic cell differentiation. Now, the other thing that's of interest to me is Ebola virus infection. And one of the recent publications we have is understanding using how do we use antimicrobial peptides to inhibit Ebola virus. And so these were the projects that were ongoing in my lab. And then what happened was coronavirus, like we all are familiar with. And so just as a brief timeline and without getting into the specifics of how coronavirus works, just to let you know as an introduction, we know that in late 2019, there were several cases of a severe acute pneumonia that occurred in the Wuhan province. And just to fast forward after that, it was identified that there was a link to a Wuhan seafood market. Fast forward as it relates to the United States, we knew that in January 20th, countries outside of China reported their first cases. And in February 28th, which happens to be my birthday, the United States confirmed its first COVID-19 death. Now, what we know right now, as it relates to the U.S., is that as of yesterday, we have 3 million cases, 3.5 million cases, and a total of 137,000 deaths. And so what does that mean? Well, what that means is that we then decided that we needed to prioritize working on this pathogen. And so my lab was tasked with trying to investigate and understand it. And so what I would like to do is go through a few of the different projects that I've been working on. And please feel free to ask questions as I go along. And the idea of what I'm trying to do is let you know exactly what we're doing, so that if there's anything that's of interest to you guys listening, you can reach out to me and ask more questions about it, or figure out a way to collaborate and be a part of the process of what we're doing. So the reason the project and the reason the title was called Inside and Out is because I've been working on both understanding transmission of the virus in the environment, as well as the molecular mechanisms of how the virus works. And then within that, also looking at possible inhibitors. So what I'll go through with you are the aerosol transmission studies we've been doing that Inakshi alluded to, as well as I'll show you some data about our attempt to look at different inhibitors, as well as a human bronchial epithelial cell model to try to understand how infection occurs. And then I'll just do a brief one slide about macrophage infection, and then a little bit about wastewater and viral surveillance. Okay, and so just to start off, we don't have a lot of data, but this is an interesting project that we've just started. And what we're doing here in Omaha and Lincoln, Nebraska, is using wastewater to track COVID-19. And the way we do that is just because we know that you can sample wastewater to identify and utilize it as a way of doing surveillance for infections. And so what this project is looking at detection of the virus and detection of viral RNA in municipal wastewater. And what you can do is you can correlate the concentration of that to the detections of the wastewater samples and virus that's in the community. So this is a project that we've just recently started. We don't have any real data for it, but this is something that I really wanted to mention because this is something that I think is translatable to other communities as well. You can identify the amount of virus present in the environment by just simply sampling it in wastewater. So this is one of the projects that we're initiating. The next project that we've initiated, and this is with the in collaboration with the Gendelman Lab, is to understand the infection of macrophages. And this is really key because we know that a lot of the deaths come from what's called a cytokine storm. And we know that there's an overreaction of the innate immune response and then the adaptive immune response. And macrophages play a key role in that. So what we've been trying to do, and what I'm showing you on the image, is our first efforts to infect macrophages and try to see if they can actually generate a productive replicating virus. And why do we want to do that? We want to do that because we want to know the effect of replication in these cells, and we want to try to mitigate the infection. Macrophages will play a key role in the response to infection, and macrophages will play a key role in how the infection occurs. So in collaboration with Dr. Howard Gendelman, we're trying to set up these initial studies just to first see how we can get macrophages infected, whether the virus will induce a productive versus an abortive infection in them, will it actually replicate, and exactly how that process occurs. So those are the two initial stages of projects that I'm starting. Now, what we're gonna get into, the next step is what I've been working with Dr. Todd Wyatt, and what we're looking at is human bronchial epithelial cell infection. And to do that, what we're really trying to do is to use air-liquid interface cultures. And air-liquid interface cultures is a way in which you can get cells to stratify. And what we want to do is understand when they're stratified in the lung epithelium, exactly how they can be introduced to the virus. And one of my PhD students, Ope, has been working on that and looking at that. And with Dr. Todd Wyatt, one of the great things is that he has available to him, because he works at the Veterans Health Administration, he's able to get healthy lungs and different kinds of lung donor cells. And so we can get these different lung cells, these different epithelial cells from the lung, and we can stratify them and put them in air-liquid interface culture. And then we can understand, are they more permissive to infection, less permissive to infection? is there more of an immune response in these particular cell cultures? And the idea is to prepare these cultures and characterize them in a disease state. And so we want to understand if you infect a lung that is from an older patient or someone with COPD, chronic obstructive pulmonary disease, or a patient that has smoked a lot of cigarettes, or a patient that has alcohol abuse. If we can look at those different air-liquid interface cells and see differences in them, then can we actually figure out the best way to treat them? And so to start that, one of the techniques that we use in my lab is the operetta high content imager. And it's something we have from this company called PerkinElmer. And it's basically a multi-parametric fluorescence-based system. And so it's a 96 well plate format. We plate cells, we then will pre-treat them with compounds, or we will do any other kind of perturbation to the cells. After that, we will then infect. And then after we infect, we then stain for viral antigen. And what you're seeing on the left side where the positive infection is what we would see as a positive viral infection, and the right side would be a negative infection. And so doing that, we were able to first try to identify whether these human bronchial epithelial cells are actually permissive to infection. And if you look on the left-hand side, you're going to see primary human bronchial epithelial cells from a patient that is older, that was cultured. And you see the uninfected cells versus the infected cells. And the red is the viral antigen that we're able to see. And then on the right-hand side, you see immortalized human bronchial epithelial cells, again, uninfected versus infected. And we use the operetta high content imager to try to see that we can actually get infection. This is before putting them in air-liquid interface. This is before the cells are stratified, because one of the things you want to do first is understand the permissivity of these cells to infection. And sometimes the cells are not able to be infected right away. You need to get them stratified. But right away, we're able to see that these cells, in fact, are permissive. Now, the next step is to understand how they work in ALI. And again, this is an experiment that Ope did. And what we're able to do, and if you follow me, if we go back, when you look at the air-liquid interface, if you look at the air side, the cells are stratified. So you have your apical surface, your basolateral surface. And what we were trying to understand, what Ope was trying to do, was understand when the virus actually infects the cells, where and how is it released. And so what we did in this experiment was we were able to do, Ope did a 1 to 5 day infection, and just pay attention to the apical graph on the left-hand side. And this is a PCR reaction, so we're looking at cycle numbers. And so what we're seeing is that we're comparing human bronchial primary epithelial cells in ALI, air-liquid interface, from a young donor and from an old donor. Ope infected with an MOI of 0.01. And what you can see is that in the apical side, you can see that there's a level of infectivity going on from day 1, day 2, day 3, day 4, day 5. Importantly, if you look at the basolateral side, the CT values or the CQ values, you see them there at about 40, which means there's not a lot happening. There's not a lot of virus being made. And then if you look further on the right, you see that in the cells, within the cells, the CQ value or the CT value is a lot lower, which tells you that the virus is replicating inside the cells. The virus is budding from the apical surface of the cells, but it's not budding from the basolateral surface. So we can see in this air-liquid interface culture from the human bronchial epithelial cells that the virus will specifically choose to replicate inside, and then for the most part will bud from the apical side and not the basolateral side. This is an important fact to understand because different viruses will do that differentially. And so what we're trying to understand is exactly what makes and allows for it to move this way. So this is one of the projects that we're doing in collaboration with Todd Wyatt. And one of the other things we're doing, and we've put together a grant to try to understand it. And essentially, we have access to this lung transplant biorepository to receive lungs from donors, and we can get the donor history. And one of the things we want to do is examine primary bronchial epithelial cells, and we want to be able to see if they can be ciliated, and if the ciliation actually can affect infectivity and how that will actually impact how the virus can get into those cells and then spread from those cells. And so what we want to do, as it says on the third point, is determine if SARS-CoV-2 infection can produce greater cilia or greater cilia slowing, or can it impact cilial detachment in epithelial cells? And how does that actually impact the spread of the virus in these particular cell cultures? And the next thing that we're also interested in is using antimicrobial peptides like surfactant D that these ciliated cells will actually make as well, to see if they can play a role in inhibition. So this is another one of the projects that I've been doing in collaboration with Dr. Todd Wyatt. Now I'm going to go through a couple of inhibitor studies that we've been working on. One of them is in collaboration with Dr. Gus Wang, and that's using engineered antimicrobial peptides. And what this graph is showing you is, if we look at SKI1, which is a sphingosine kinase inhibitor, don't worry about that one, DI128A, L101, and L102, and L102, these are antimicrobial peptides that Dr. Wang has engineered. And what we're showing, if you look at the y-axis looking at percent inhibition, we're able to show that we can get greater than 90% inhibition using one of his antimicrobial peptides against SARS-CoV-2. The way this experiment works is that we treated Vero E6 cells with 20 micromolar of the antimicrobial peptides for 1 hour. Then we infected with SARS-CoV-2 at an MOI of 0.01 for 24 hours. And again, we stained them. And so these are one of the inhibitor studies we've been doing, looking at antimicrobial peptides. In collaboration with Dr. Jonathan Vennerstrom and Dr. Caroline Ung, we've also explored the potential of using anti-malarial compounds. And what you're seeing is an experiment in which we've used bisquinolones as a potential novel SARS-CoV-2 antiviral. And on the top panel A, we've treated these cells with these different bisquinolones and in Vero E6 cells. The bottom panel, we treated these bisquinolones in HepG2 cells, which is a hepatic cell line. Again, we did 20 micromolar treatment with the indicated compounds, and we're able to see that we can get, you can see with TP37, for example, we're getting almost 100% inhibition of the virus. And with TP37 in particular, between HepG2s and Vero E6 cells, we're getting very high inhibition. So in addition to that, any questions, by the way, while I'm going on? Any questions? Somebody say something.
No, sir. You can continue after your talk is over. There is a question answer session. Some questions will be there in the YouTube chat box. So someone from us will convey the questions to you.
Okay, cool. Okay, so in continuing with just showing a couple of the inhibitor studies that we're also conducting, is we're also looking at the possibility of whether known antimalarials can play a role. And this is a grant that we're working on right now. And again, this is in collaboration with Dr. Jonathan Vennerstrom at UNMC and Dr. Caroline Ung. And what we've been able to do was to show, and if you look at the graph, you'll see there's CQ, which is second from the right, that's chloroquine. And we can show that there's about a 70% inhibition against SARS-CoV-2 with chloroquine. Hydroxychloroquine, which is HCQ, gives a greater inhibition. But you'll notice that like antimalarials like methylene blue can also play a role. And mefloquine as well, which is the MF, you can see that that's a greater than 90% inhibition. So what we're doing right now is trying to understand the role of these different antimalarials, the mechanism in which they're able to block the virus. And again, this is in collaboration with these two labs. And so what we're really trying to look at is ozonides, diarylureas, and bisquinolones, because they're an untapped chemotype against emerging coronaviruses like SARS-CoV-2. And what we're trying to do is develop a program to understand how they work. And so in collaboration, again, we're going to synthesize these target compounds and we're going to characterize them. We're going to characterize them by looking at the in vitro activity against the virus. We're also going to look at the cytotoxicity, the solubility, the permeability of the cells, and this is the admin properties. The next step is we're going to understand and try to assess the pharmacokinetic properties of the selected target compounds. And then we're going to investigate the mode of action or the mechanism of action. Exactly how is it that those specific antimalarials inhibit the virus? So this is one of the projects we're working on that I'm very, very excited about. And now the next thing that we're working on with Dr. Benson Idagwa is looking at niclosamide. And again, this is another project that my student Ope did, is to look at niclosamide, which is a federally and FDA-approved antiemetic drug. And what you're looking at is on the top upper part of the screen is the green in the upper left-hand corner is what the infected Vero E6 cell looks like. The right next to it is the uninfected cell. And what we did was we treated with different concentrations of niclosamide. And you can see we went from 100 micromolar all the way down to 0.1 micromolar. And you see that it goes from less green to more green, which shows that the infection starts to increase when you use less of the compound. And what we're able to do was use a dose response to show that we can get an IC50 of about 1.1 micromolar of this compound. Why it's important for us to do this is Dr. Benson Idagwa is a leading researcher in nanoformulation and long-acting drug activity. And so he can make a prodrug of this and then allow for it to be long-acting in the cells. So these studies are just to first and foremost show that niclosamide can inhibit SARS coronavirus 2. The next step will be to formulate it and then allow for it to be long active as a nanodrug particle. So I'm excited about this one particular data set that we've been working on with Benson. So that's the compound study. Now, the next part of it, and this will be the last group of data that I'm going to show you, and this is something I've been working on in collaboration with Dr. Josh Sartarpia. And so what happened is, at the University of Nebraska, what you can see in that image is the Global Davis Center. The University of Nebraska was charged in early March with caring for 13 individuals who came off the Diamond Princess cruise ship. They were admitted to the University of Nebraska's biocontainment unit. The mildly ill individuals were isolated in the quarantine unit, and the individuals who needed hospital care were in the biocontainment unit. What I did in collaboration with Dr. Josh Sartarpia was we then looked at whether we could identify evidence of viral shedding in on the surfaces and in the air in those different areas. And so within the study, we used and we detected basically high volume air samples, low volume air samples, and personal air samples. So the individuals who went in the quarantine units had their own air samplers. Surface samples were collected from the common rooms of where individuals were, the surfaces, personal items, toilets, also the personal air samplers worn by the individuals. We sampled, and then what you were able to do was do qPCR or RT-PCR on those air samples. And of the 163 samples that were collected within that, in the biocontainment unit and the quarantine unit, 121 of them were PCR positive. Now, what that means is that we're able to just identify the fact that there's shedding of RNA in all of those different spaces. Now, that doesn't imply that there's actually infectious virus there. What it means is that there's RNA present. So we then endeavored to look and explore where exactly those samples were. And so on the left panel A, what we're showing you is the copies per microliter of recovered samples or recovered positive PCR samples. And this is just to give you guys a snapshot of where we actually looked. So Josh and his team looked at bedside tables, air handling grate, the floor under the bed, the room window ledge, phones, the remote control that they were using to watch TV, the toilet. All of these were PCR positive. and then there's bedroom air samples. The panel on the right, which is panel B, shows the percent positivity of those different samples. Now, the next step, once these are PCR positive, is to know whether this actually will produce infectious virus. So what we did was we were able to take the gelatin slabs that came out of these different air samplers, and we tried to culture them. And what we're showing you, and what I'm showing you now, is one of the things with coronavirus, which is similar to a lot of other viruses, that if you allow for them to infect over time, you will then get CPE, and you'll start seeing cell death, which is shown by the left-hand side in the NQU hallway air day 8 and the NQUA window cell day 5. These cultures were grown up over 5 days compared to your control Vero E6 cells. And what you're seeing is the virus starting to actually induce what's called cytopathic effect in those cells. So we were able to take air samples, hallway air samples, and window cell samples, and show that these samples, in fact, have infectious virus. Another way we're able to show that is by doing immunofluorescence and immunomicroscopy. And again, what we were able to do was to take the hallway air day 8 samples, put that onto cells, and then stain for viral antigen. And in the IFA that you're looking at that says panel E, is we stain for the SARS coronavirus spike protein. And what you see, especially in the lower right-hand corner, is you can see green fluorescence, which indicates that there's an actual active replication of the SARS coronavirus spike 2 protein. If you look at the immunofluorescence image, the control, there is no green. And then if you look at the EM, we took Vero E6 cells from those cultures as well and did EM. And what you can see are prototypical viral particles inside of the Vero E6 cells. So what we're able to show is that from these PCR positive samples, you can in fact culture virus out. And it means that it's not only that there's RNA present, but within these cultures, there is active virus replication. Now, what this means is that there's environmental contamination in the rooms where these patients were infected, and it seems to be regardless of the degree of the symptoms or the acuity of the illness, meaning that there are individuals that were not symptomatic with infection, but they were shedding virus in a way that we could detect it. And again, just to underscore, so it means contamination exists in all types. So we're able to get contaminated viral samples in the air, personal items, room surfaces, and toilets. And so this was the first manuscript that we just produced. And right now we're working on a second manuscript. So in this initial one, what we were looking at is aerosol and surface transmission. The next step was to look at simply aerosols and different kinds of aerosols. And so what we endeavored to do was investigate the infectious nature of SARS-CoV-2 in droplet and fine mode aerosols from 6 individual patients that came into the biocontainment unit. And so what we did here was just simply collected air samples, and we collected air samples in a size fractionated pattern. So only collected particles in less than 4.1 micrometer versus 1.4 versus less than 1. And so these are just air samplers that are able to trap different particle sizes. The reason why we wanted to do that is to look at the droplet sizes versus the aerosol sizes. And we know that there are these different fine sized particles. So if you're able to collect them in these different sizes, then you can then say, if you can trap them and culture virus out of them, you can say if it's an aerosolized particle versus a droplet particle. So we collected particles in these different sizes. And what we're able to show is, first and foremost, if you just look at the black bar on this graph, is that we're able to show that you can get PCR positive samples from these 6 different rooms that we collected from. What I was able to do was, from the particles, culture them again and grow them up over 9 days, and then show that you actually have actual replicating virus. And what this is a Western blot looking at SARS coronavirus 2 N protein. And so we're seeing the production of N protein in samples A, B, less so much on B, more so much on C, but on samples D and E, you can clearly see that we're getting the production of the N protein in these cultured cells, meaning that we're actually getting virus making more proteins, meaning we're getting infectious virion production from these cultured samples, from these aerosolized particles that we've trapped. And again, we did another EM study to show that we're able to identify the presence structurally of these virion particles in that space. So again, I wanted to share this data with you guys so you know the work that we've been doing, and that would be it. So in the end, what we did was we showed that the infectious SARS-CoV-2 aerosols can be detected, and they were primarily observed in the less than 1 micrometer fraction of the gelatin filter that was designed to actually collect intact virus. What we're going to do now is continue further studies to identify the particle production over time and understand the sequences between the different viruses that are being released by individuals. We're also doing stability studies to understand if the virus is being released, is the viral RNA stable over time? Is it a matter of just the RNA being there, or does it need to be the actual viral structural components? And can you get replicating virus over time from these individuals that are expiring particles? So with that, I will say, thank you very much for listening to me. I would want to acknowledge first and foremost my lab that consists of Jane Mordser, Inakshi Roy, and Ope. And Ope did a lot of the studies that we're doing. And by far, none of this is done by myself. Most of the work that I'm doing is in strict collaboration with a number of individuals. I would like to acknowledge Todd Wyatt, because Todd Wyatt has been working on looking at human bronchial epithelial cells for a very long time. And we've just translated that work into understanding the way the virus might infect these cells. Howard Gettleman has been working on macrophages and the HIV sphere for a very long time. And so we've now started collaborating to understand the role of macrophages in SARS-CoV-2, the cytokine storm that we know exists, whether there is an actual productive infection in these macrophages, whether it's an abortive infection, and the role that macrophages play in disseminating, spreading, and being able to utilize it. Can we utilize it to treat the virus? Dr. Jonathan Venestrom and Dr. Caroline Ung are leaders in the field of anti-malarial research. And what we've been trying to do is utilize anti-malarial compounds to see if they can also masquerade as anti-SARS-CoV-2 inhibitors as well. Dr. Benson Adegwa is working on the NCL study. And so what we've been trying to do is utilize his forte in looking at whether this nucleoside can actually work as an inhibitor, not just for it to work, but because it's a nanoformulated particle that can be long lasting. It might be something that you can administer to someone now, and it will stick around for months to years and actually have an effect. Dr. Gus Wang, his lab is close to mine. He's been working on antimicrobial peptides for a very long time and utilizing them. And so we started working on antimicrobial peptides and showing that they can actually inhibit Ebola virus. So it was a natural movement to see if these same antimicrobial peptides that the body makes can actually work against SARS-CoV-2. So far, we've seen that it can. Josh Santarpia, his lab as well, has been helping, and we've been doing the aerosol studies to understand transmission, expiration. I didn't have time to get into work that I've been doing with Dr. Dalia Elgamal on bromodomains and epigenetic studies on how the virus works and identifying whether you can target bromodomains using TVET inhibitors. Dr. Sadapa Bayareddy is someone else that I've been working with to look at kinase inhibitors and whether protease inhibitors can work. There are also collaborations with Far Biotech and Helion Bio, where we're doing a number of screening studies. And with that, I would thank you for your time and thank you for listening. What I tried to do is give you a very broad brushstroke of the kind of work that we're doing. In the event that maybe there's something we can collaborate on, there's a way that you can help, there's a way that I can help. These are all the different capabilities of the lab. This is what we're doing. And I will welcome any and every question you can ask. Thank you very much for listening.
Thank you, Dr. Reed, for an engaging presentation. Here's looking forward to a great interactive session ahead. For that, I invite Satvika Das, a fourth semester postgraduate student of the Department of Microbiology, Vijaygarh Jyotish Ray College, to conduct the question and answer session.
Thank you, Harsha. I would take it over right now. First of all, sir, I would like to tell you that it was a very, very well enriched session. And now I would like to present some questions that were asked during your presentation. So the first one is from Inakshi Roy. She asked a question. The MOI 0.01 is quite low. So is that an indication that a lower number of virus particles can infect the lung cells in vivo conditions?
So the MOI of 0.01 is relative because that determines how the virus will infect and replicate. It's not actually low, it's relatively low. We know that in our studies, if you take an MOI of 0.01 and infect a certain number of cells, we can get a, let's say, positive infection over a 24-hour period. That's different from an Ebola virus infection where if you took Vero-E6 cells, again, so let me take a step back. In 2D culture, which means when you put Vero cells on a plate, that's very different from what happens in the body. And viruses do not respond the same way in the body as they do in tissue culture. You can put Ebola virus on Vero-E6 cells and you will require an MOI of 10 to 50 over a 24-hour period to see a positive infection. Venezuelan equine encephalitis virus, you can do an MOI of 0.0005 for 12 hours and see a positive infection. It's an artificial surface that we're creating, and so what we're trying to do is understand when and how the virus replicates. So with all that said, an MOI of 0.01 is not actually low. It's relatively low compared to how the virus actually will replicate. But what you do in tissue culture does not at all and in any way recapitulate what you see in vivo. So this is all relative numbers that really literally mean nothing. So what you're trying to do in tissue culture is see how well you can see positive infection. You don't extrapolate that to mean a low or high MOI. You just know that that's what you see in tissue culture. Did that answer your question, Inakshi Roy?
Yes, thank you.
Okay, sir. Moving on to the next question, which was asked by a faculty, Dr. Gargi Shahakesh. Why do you think is the reason that antibacterial peptides are inhibiting SARS-CoV-2?
So those are antimicrobial peptides, and more than likely what they're doing is they're playing a role in the cells and not targeting the virus itself. What we know from other studies is that if the cells can be primed to respond to an infection, they can be refractory to that infection. And so antivirals or antimicrobials or any agent that you're going to use against a pathogen are going to do one of two things. They're either going to target the pathogen specifically or they're going to target the host itself. And sometimes what you'll do is you can trigger a response to the host that would allow for it to be refractory. So I would actually posit that you actually want to target the host and not target the virus, because then you can have something broad-based and long-acting. And so in answer to your question, sir, it's because it's targeting the host.
Thank you, sir. In relation to this, sir, we have a question from Meghna Nandi. How do you determine what kind of compounds to use for inhibitor studies?
Whatever I get from people who want to collaborate with me.
Okay, sir. Another question.
I don't make them myself because I'm not a medicinal chemist. Medicinal chemists create them, and I will try whatever compound you have.
Okay, sir. There is another question. Actually, there are a lot of questions, and I'm trying to filter them the best I can. Why is hydroxychloroquine better than chloroquine at inhibiting SARS-CoV-2? This was asked by Devdatta Das.
It's not. It was in very easy.
Yes, sir. I think there has been some miscommunication because it was clear in the graph that chloroquine had a better inhibition.
No, it wasn't, and it didn't.
Yes, sir.
It's
Okay.
Vero E6. First and foremost, it was Vero E6 cells, which are not real cells. These are experimental cells that we're looking at and seeing how they work. These are monkey kidney cells. So the inhibition you're observing is relative. You don't think of it as an absolute, because what's going to happen is we took one concentration over one specific period of time in a 2D cell culture model. And what you do is you understand, relatively speaking, what that means. You don't take it too far. What you do with that information is you say that these compounds have activity. What you don't say is that one is better than the other.
Thank you, sir. There is another question. It has been suggested that the chloroquines can change the acidity at the surface of the cell. It can also activate immune system, thereby preventing the virus from infecting it. But how exactly?
So the simple answer is what we're talking about, and what the way it works, as far as I understand it, is that when the virus infects the cell, it requires something for entry. So when the virus, so SARS-CoV-2 has to bind to ACE2. It then, after binding to ACE2, it needs to be processed. And when it gets processed, it requires a pH-dependent event for that to occur. What these compounds, like chloroquine, will do is mitigate that acidification event that occurs in the endolysosomal compartment. In that way, it blocks the ability of the processing step to occur. That's what I believe is understand. That's what I believe is how this works. So it's not that there's a change in the outer part of the cell. It's the endolysosomal compartment that gets deacidified. Does that make sense?
Yes, sir. And there are a few more questions I'd like to ask. So,
Yes.
two of the questions are there, sir. I'm merging them. It was like in the content, you have said that MF or MB, the anti-malarial compounds, they have a high rate of inhibition. So why aren't we using them? And another person has asked, how close are we to an actual vaccine?
Good question. Those are two really good questions. So the answer to the first question is, what we have to do after showing inhibition in Vero E6 cells is then showing inhibition in an actual animal model for infection. In answer to your, so then we would have to show that the compounds can inhibit in an animal model. The next step we would have to do is what I outlined with our studies with Dr. Caroline Ung and Dr. Jonathan Venestrom, is we need to do pharmacokinetic studies to understand the bioavailability, the ability of these compounds to be in the body and actually work efficiently. So these things require us to understand the chemotype of the compounds and a lot of other factors. So there's a few other steps that has to go once you know that they can be inhibitory in cell culture. Now, in answer to your vaccine question, I would say to you that you should ask, do you need a vaccine? And the reason I ask that is, what does a vaccine require? And how long does the immune response come from a vaccine? And I would tell you that for different viruses, you will get different kinds of immune responses. And so I would posit that for this particular virus, a vaccine is not going to be useful. Small molecules, antivirals, targeting the host cells to inhibit the virus is going to be more useful, because if you are to then get a vaccine, you're not going to get a long-lasting immunity. That's my opinion.
Sir, should we take a few more questions?
Yes, please.
Okay, so there is this another question. How are you considering recruitment of various cell surface receptors along with ACE2?
That's actually a very very good question. So I have not looked into that. but there is always a possibility that there are other cell surface receptors that can play a role in entry. We know, for example, with a number of viruses that, for example, lectins or your carbohydrate binding molecules on the surface of cells will play a role in just allowing for the viruses to attach. And they may not mediate entry per se, but they'll allow for attachment. And so we know that with a number of viral examples that it's never just the one receptor, and binding is just not one-to-one. So that's a very good question, because we know for a fact in a number of examples that it's not just that initial binding by that one specific receptor. For Ebola, for example, it's not the Ebola receptor is actually inside the endosome. So it's not actually binding to the cell surface. Ebola actually gets engulfed by macropinocytosis and then gets opened up, then it binds its receptor inside the cell. So we know from various examples that there's going to be multiple factors that can play a role in binding and entry. So that's a very good point. And so we don't know that there's anything other than ACE2. I would endeavor that the person that asked that question should work with me to try to find the other receptors.
Okay, sir, I think that has reached them and maybe they'll try. So, sir, there's another question. In the ALI studies, will you be able to use lungs from COVID-19 patients and screen the most promising drugs from your inhibitor studies?
That would be something that we would really like to do. At this point, though, what we know from the lung bank is that we're able to get samples from individuals who are veterans. And the thought initially was just to get different kinds of bronchial epithelial cells from individuals to show whether an older bronchial epithelial cell versus a younger bronchial epithelial cell is more or less susceptible to infection. And if that susceptibility is associated with the ability of those cells to be ciliated. And then if those cells are ciliated, do they produce enough mucus, and are they refractory to infection in that way? So the initial studies were just to try to understand mechanistically and physically how those cells work, and whether we can see that individuals are more or less susceptible, and is the bronchial epithelial cells that are gotten from smokers or someone with chronic obstructive pulmonary disease, if they're more or less susceptible. Now, in an ideal setting, we'd want to actually get lungs or bronchial epithelial cells from someone who was positive. But the reason you would want to do that is to identify whether those individuals, if let's say it was someone that was positive for COVID-19 and they had an asymptomatic infection versus someone who had, let's say, a more severe infection, if we're able to take those lung cell cultures, those bronchial epithelial cells, and identify anything about them that are different after the effect of the virus being there, that would be an interesting study to do. But you don't want the cells from those individuals now. You want cells from individuals to understand the impact of the virus on them. So you don't want it from COVID-19 patients. You want it from individuals who are either smoking, not smoking, drinking a lot of alcohol or not, because what you want to do is try to predict in the population how they will be impacted in the future. So you don't want individuals that are sick with COVID. You want to understand from individuals who have not seen it yet what might happen when they do see it, in order to then try to mitigate it by understanding what their susceptibility is to it. And then what can we do in advance of that to try to prevent them from being more or less susceptible.
Thank you, sir. That was a wonderful explanation. So in relation to patients and recovery, I have two questions which I would like to pile together. So can you enlighten us about how the cytokine storm caused increased severity for SARS-CoV-2, and recently no antiviral drugs are made, but somehow patients are recovering from SARS-CoV? Can you kind of explain that or shed some light on that?
So what little I know about the cytokine storm is that essentially what happens is there's just an overproduction of pro-inflammatory cytokines, and that's usually mitigated. That's usually actually initiated by your immune cells, your macrophages, for example, secreting tons of your pro-inflammatory cytokines, and I'm not an expert by no means on it, so what I would not like to do is provide any disinformation. What I know is that there's a pro-inflammatory response that cannot be mitigated, and that's a part of what the cytokine storm is. The next question about antivirals. Can you repeat that for me, please?
Surely, sir. It was that recently no new antivirals are being discovered. So, how are the patients getting cured?
So no one is cured. A cure means something completely fixed it. What we know is that during an infection, and during every viral infection, there's two things that happen. There's a window in which the innate immune system responds, and there's a window in which the virus is replicating. And then the virus has to move from one initial site of infection to the next. With chikungunya virus, it starts with a mosquito bite and it goes to the Langerhans cells. With SARS-CoV-2, we're going into the bronchial epithelial cells. And what happens is there's a moment in which there's a battle happening, an initial response to the infection by the innate immune response. Can that mitigate the infection? Can that downplay the virus? Can that alert secreting interferon alpha and beta to tell other cells, hey, there's something wrong here, versus the virus's ability to replicate and move farther down the respiratory tract and then get into other cells like the neuronal system. Now, what we try to do and what can occur is if you can have fast-acting antivirals or different medicines that can mitigate that, and that's fantastic. What we have though is probably the fact that antivirals that are already on the market that we've already used, like your remdesivir, like your chloroquine, like your favipiravir, these will actually probably work, but they won't cure. What they will do is they will stop the virus from replicating. And when you stop the virus from replicating, you allow the immune system to then build a response to it, because you're just simply slowing it down. You're not stopping it, you're slowing it down, and then your immune system will take over from there.
So, sir, are there chances of reinfection in a patient who has already been infected by the SARS-CoV?
That's a very good question. The answer to that is I don't know.
Okay, sir. So one last question I'd like to ask you. For inhibitors, was 20 micromoles optimized or titrated? Any cellular toxicity studies performed in addition to 0E6? Any human airway epithelial cell line used or might be used in the current study?
Yeah, so it's a good question. So with the operetta, we can look at cytotoxicity by looking at whether we lose cells or not. And so we can, in the assay, always detect whether there is loss of cells or not. The 20 micromolar concentration is something that we start with. And so it's just a random concentration. So we start with 20 micromolar. We could start with 10 micromolar. But the reason we do 20 micromolar is because if you do a 20 micromolar treatment and you see an effect, then you can now start lowering the dose. If you go to 20 micromolar and you don't see an effect, then you can discard that compound right away because it's useless. And so you start at 20 micromolar, and the idea is that if you start with that concentration and you see an effect, then let's work our way down and see how low we can go. If you start with 20 micromolar and you don't get an effect, then it's useless and it has no efficacy anyway moving forward in any other samples.
Sir, can we take one or two more questions?
Absolutely.
Okay, sir.
Shatika, there is a question in Google Meet. It
Yes,
was
sir.
you should ask the question to the speaker. Okay.
Sorry if I missed it.
after you moving out from Google Meet, there is a question written in here.
Okay,
Please look
so
at that. Okay.
okay, sir. I will definitely check that out. Sir, it was asked by Nakshi Roy. Does SARS-CoV-2 produce plaques in viral plaque assay?
Who asked this question?
I asked it.
Why don't you do the plaque assay?
Well, because
The answer is yes, and I would love
actually,
for you to do the plaque assay yourself.
actually, Ope and Kavita, like they're like doing the work. So they told me that they are not getting enough plaques, and so I wanted to ask this question.
So okay, so what happens is it doesn't plaque very well, but that's a combination of a couple of factors. A couple of factors is always remember, guys, when you're using a cell culture model and you have the virus in a two-dimensional space, that's not natural and normal. A virus infects the body and it's a three-dimensional space. So what you need to try to do is recapitulate the environment it's used to the most, and then you require time for the virus to actually cause an effect, which would be to clear the cells out that you're infecting. So the first thing is, are you utilizing the right cells for the virus to clear them out? The second thing is, are you giving it enough time to do that? And so it may or may not plaque as fast as you want it to, but it's just a matter of looking at how to actually do it. The virus does plaque. The virus will replicate and clear cells. And we have plaques. We have shown plaques can work. And we can also do TCID50s because TCID50s work. Now, what we found is it doesn't replicate to high enough titers in the culture systems that we're using. And so we need to optimize that and or concentrate the virus. But again, we know the virus can plaque, it will plaque. It doesn't plaque as well as chikungunya, but it does plaque.
So, sir, the rest of the questions that we have are kind of similar, so I would pool them together for your ease. There is one question which focuses on reinfection with a different strain. There is another question which is, are the different strains of virus determining the extent of infection, that is asymptomatic, mild, moderate, and severe? There is another question that the most dangerous aspects of SARS-CoV-2 are the asymptomatic carriers. Is there any specific reason for a person to be asymptomatic? And as there are evidences of deaths of asymptomatic patients as well, that is why.
So the very last question, if you die and you're asymptomatic, we don't know that you're asymptomatic and you die, you just die. So we don't know that. We don't, unless you've been tested and you're positive for the virus and you show no signs of disease or illness, but if you die, you clearly were sick or got hit by a car or something. So I don't understand that question, to be honest. Now the other, there were a lot of questions too. The issue about strains, as a virologist, I hesitate to get into what makes a virus from a strain perspective or isotype. Like I don't know if there's a difference in terms of the actual strain that dictates severity of infection. I think fundamentally what dictates the severity of infection is the host and the relationship between the host and infection, and also the point of entry will dictate that as well. So those are the factors that will play a role in that. I don't think it's strictly up to whether there is a particular amino acid sequence change in the glycoprotein or the spike protein versus, whether there's a glycine versus an arginine in the spike protein. I think there's an interaction with the host that for the most part will dictate the severity of disease. It's less associated with the strain. I believe in most cases thus far, because this virus hasn't changed so much that there's going to be that great of a difference in the changes in the genotype of the virus. Because what that would imply is either, because it's very specific, if the spike protein changes, that would impact entry. If other parts of the virus changes, that would impact its replication capacity. Now, if we can identify that those things have actually changed, which and no one has shown that yet, what we can then say, I would believe, is then it's the impact of the host innate immune system on that and the host adaptive immune system. So remember that there are two things happening during an infection. It's the host response to infection and the ability of the virus to replicate. And so those two things will play a role.
Thank you, sir. So the last question that I'll be asking you is that the most dangerous aspect, sorry, sir, any data on proportion of exhaled droplet nuclei or of respiratory droplets that evaporate to generate aerosols and the infectious dose of viable SARS-CoV-2 needed to cause infection in another person?
That's a very good question, and that's what we're going to try to explore. The problem is, it's hard to explore that. In the studies that we were doing, we were looking in nature, we were looking at individuals expiring those particles. The way to answer the question that you asked would be to have it in an isolated setting in which you can make aerosolized droplet particles. And then you can actually see whether in those particles, when they've expired, if there is actually still infectious virions in them. Those are isolated studies that can be done. There was actually a beautiful study that was done recently where they looked at the transmission in ferrets, because ferrets can sneeze and cough, and so they can transmit as well. And they looked at the different particle transmission in ferrets, and they had ferrets that were either housed together or they had a chamber next to them and had another ferret on the other side. And they're able to show that you can actually transmit, and they can control for the size of those particles when you do those studies. So that's a very good question. And in humans, it's a really hard it's really hard to address that naturally, but that can be addressed experimentally.
Thank you, sir, for being so patient with us and then addressing all the horde of questions that came our way. So, any further?
Does Roy have another question?
Sorry, sir?
Does Anakshi Roy have another question?
For now.
Okay.
Okay, sir. So I'd like to give it over to Harsha. Thank you, sir.
Thank you, Dr. Reed and Ms. Das, for resolving the queries and wonderfully conducting the question and answer session. Now, I adjure Dr. Shilajit Barua, Assistant Professor, Department of Microbiology, to conclude this session with his vote of thanks.
Am I audible?
Yes, sir.
So, a very good evening to all our respected delegates, and very good morning to our resource person. It's my immense pleasure to welcome Dr. St. Patrick Reed to the second session of the International Webinar Lecture Series organized by Department of Microbiology in collaboration with Internal Quality Assurance Cell, Jyotish Roy College. And we are grateful to you, sir, for taking out time from your busy schedule to deliver this wonderful and robustly informative lecture in this session. Thank you, Dr. Reed. The discussion session has also been extensively informative too. I would like to thank Dr. Rajyasree Niyogi, our beloved principal madam, for her constant inspiration in carrying out such webinars. I would also like to congratulate our students, Ms. Harsha Sharma and Ms. Satyuka Das, for conducting the session in a beautiful manner. It's a very proud moment for all of us to have among us one of our ex-students, Ms. Enakshi Roy, and we would like to wish you all the best for all of her future endeavors. Next, I would like to thank Dr. Prosenjit Das for the constant technical support. I would like to thank all my colleagues, students, and all of the distinguished participants of this session. Thank you.
Thank you, sir.
So the session is now over. Congratulations to Inakshi for cooperating with my request, and obviously heartiest welcome to Dr. Reed. So we will keep in touch in near future definitely. So best wishes for your research endeavors. Thank you.
Thank you very much for the invitation. Thank you very much for training Inakshi and making her the great student that she is. And all my job is to do is to make her even better, that I can send her back to be an even greater scientist. So thank you very much for what you've done for me in allowing for me to meet her and for my being able to play a role in developing her into this great scientist. It's a pleasure. And thank you for the invitation.
Thank you, sir. Thank you. Thank you very much.
Okay, I'm leaving. Goodbye.
Okay, sir. So, Akira, So, thanks to Harsha and Shatika, you have done wonderful. Please now leave the meeting. We'll have a little chat with other members of the faculty.
Okay, sir. Thank you, sir.
Thank you. পরে কথা হবে।
Okay, thank you guys. Bye.
Thank you. Thank you. সো এবারেরটা মোটামুটি ঠিকই হইলো নাকি?
আমার ঘাড় মুড়ে যাবো সকাল থেকে আমার
হ্যাঁ আমি তো মানে বসে থেকে থেকে
এবার ঘুমিয়ে পড়ো না তুমি
সিন নেই আমার খাওয়া যাচ্ছে অনেক